This presentation is on the investigation on the developmental biology of Vanessa Cardui by Andrew Sanchez, Michael Beard, Paulina Amaton, and Beyonce Macias. Arthropods are invertebrate animals that have an exoskeleton, which is a rigid external body that supports and protects the animal. The study of arthropods is important because many arthropods can be dangerous to humans and affect our health and well-being. They also affect us in ways like destroying crops of fruit that we grow. They're necessary as a food source in the food chain, and some arthropods are even eaten by humans, for example, crustaceans like lobster, crab, and shrimp. There are benefits as well to understanding arthropods, for instance, honeybees and butterflies. In this lab, we attempted to further understanding on the growth of a particular arthropod, the painted lady butterfly. We studied two different larvae and compared their growth rates, as well as what could affect their rate of growth depending on the amount/duration of light. Vanessa Cardui, also known as the painted lady butterfly, is a species within a group of arthropods known as holometabolous insects. This group refers to insects that undergo a complete metamorphosis. This unique development seen in other insects such as moths, bees, and beetles is a process of transformation from an immature stage to a distinct adult form. Metamorphosis commonly begins with the larval stage, or the caterpillar phase of the insect life cycle, before continuing into the pupal or cocoon stage. After a certain amount of time has elapsed, the insect will hatch and emerge as an adult butterfly. The painted lady butterfly inhabits almost all of North America, and it's widely known for its scattered and quite large migration patterns. Vanessa Cardui, meaning butterfly as thistle, is classified under the Lepidoptera order of insects that includes butterflies and moths and the Nymphalidae family. The Nymphalidae family is the largest family of butterflies, encompassing over 6,000 species. The life cycle of this cosmopolitan butterfly begins with a spherical-shaped, green-colored egg often laid atop a small leaf or branch. From this egg, a caterpillar will hatch and feed on surrounding leaves and vegetation, continuously growing in length and slowly changing color. The larva will molt 5 times before producing a silk button on which it will suspend using its prolegs. The caterpillar will assume its characteristic J shape before moving on to the pupal stage. The pupa stage lasts approximately 5 to 8 days before an adult butterfly with white dotted orange wings, brown and gray color patterns, and a signature proboscis emerges. The purpose of this investigation is to understand the life history of Vanessa Cardui and explore the effects of environmental changes on its growth and overall life cycle. To do so, painted lady larvae will be grown under differing environmental conditions and monitored on a daily basis for any physical changes, including weight, length, and overall appearance. For this experiment, a control and an experimental group was designed based on exposure to simulated sunlight. The control group of caterpillars will receive 12 hours of light and 12 hours of darkness to simulate the day and night cycle as in the wild, while the experimental group will be observed only under darkness. The amount of food given to both groups remained constant at approximately 6 grams. We hypothesize that a total lack of light will have a negative impact on the painted lady's initial larval stage and results in stunted growth.
The materials needed for this experiment include 2 plastic containers, some larvae food, a ruler and metric, a weighing boat, a weighing scale, 2 larvae for each group, a single spoon, and some napkins to place these larvae on. As you may recall, our larvae were split into 2 different groups, the control being that they were grown under 12 hours of light and 12 hours of darkness. In contrast, the experimental group was grown under 24 hours of complete darkness. Each and every day, measurements will be taken of these larvae. To begin taking the measurements, we will use the spoon to carefully remove the larvae from their container and then go ahead and place it on a napkin. It is very important to be careful in this step, as at the very beginning of the investigation, the larvae are very small, so it is important not to harm them. After placing them on the napkin, we're going to go ahead and get a ruler, measure them in centimeters, see their length. Upon measurement, we'll go ahead and place the larvae on a measuring bolt that will then be placed on a measuring scale. At the beginning of the investigation, the larvae may be too small to measure, as their weight will be read as just 0 grams. However, later on as their growth is measured, they will eventually start weighing more and more. Once all measurements have been taken, we will once again carefully place the larvae back into their designated containers, again being very careful not to harm them, and go ahead and place them under their conditions once again. These steps will be repeated each and every day that measurements will be taken. On to our discussion. Once again, our 2 larvae for each group were measured each and every day. The amount of food was constant at about 6 grams and was only changed if needed, whether the food went dry or if the specimen ate up most of its food. Once again, our control group was the amount of light. The control group only had 12 hours of light and 12 hours of darkness. And in our experimental group, it grew under 24 hours of complete darkness. Throughout this week, we were able to analyze the size and development of these larvae and eventually compared both at the end of the experiment. As mentioned in the results section, the control group did perish sometime over the weekend, and after looking over at the final day of its measurements, we saw that it had almost no growth from the beginning of the investigation, as it still did not weigh on the scale and kept about the same length. As for our experimental group, it had a final length again of 200 millimeters and a final larval weight of 0.1 grams. With this said, it can be inferred that our experimental group of 24 hours in darkness does indeed contribute to the growth. In doing this result, we have concluded that our hypothesis was proven to be incorrect, as again, the experimental group did manage to outgrow the control groups. As for some determining factors on why our control group could have deceased, the main one thinking could have been cannibalism. As we had said, we did lose one caterpillar. Perhaps our chem wipe was not tightly secured around the air holes. As we had mentioned, cannibalism is pretty abundant with these species. Other than that, perhaps the amount of light had overheated the container, and perhaps the specimen had died due to overheating.
For our larva insects, which are called Vanessa larvae, we measured them every day and changed the amount of food we gave to the larva. We made sure that the food wasn't dry and it will not affect the larva's growth. We used light as our independent variable. We used it to manage if it didn't have any effect on the growth of the caterpillar. Through time within a week, we analyzed our larva size and development. We compared our results with 2. We had our control independent. For our control, we observed and kept the conditions of our larva by exposing it to natural light and small light bulb. As time passed by, we saw our results. For our dependent conditions, we had the larva within the 24 hours of darkness, and we saw the different effects.
Results. The growth that was presented by the 2 larvae was phenomenal. The larvae were growing at a fast pace, taking into consideration the environment that was presented. Each day, the larva would grow at least 2 or 3 centimeters, as it is shown in the table. Both larvae looked extremely healthy. They were moving at a good pace, moving up in the containers they were each assigned to. However, since the larvae were not in the same environment, as time passed by, the changes presented were noticeable. The dependent larva, which was the one that was in the dark the full 24 hours, grew at a much faster pace than the control. It seemed that throughout the entire experiment, the control larva grew, but not as much as the other one. The control larva was showing good movement the first few days, but as the experiment continued, the larva just barely moved and shrunk, as it is shown in the table presented. Unfortunately, by the 5th day, this larva was dead. But the dependent larva was very healthy and kept growing at an amazing pace. Conclusion. In conclusion, our results did not correlate to our hypothesis. We assumed that due to the amount of light or lack thereof, the dependent larva would have considerably less growth, but the opposite turned out to be true. Our controlled larva, which was exposed to more light, did not grow, but in fact, it died. Our dependent larva grew a decent size considering the amount of light it received. This definitely did catch us by surprise, as we expected a different outcome. We do not know why or how this happened, but it is interesting knowing that the less light the larva was receiving, the larger it was growing.